Friends, welcome to Biology Made Easy. Today in this video, we'll discuss about the cell wall. Now this cell wall is the basic difference between plant cells and animal cells. That means the cell wall is unique only to plant cells. They are completely absent in case of animals. And a cell is actually surrounded by certain cell walls. This was first observed by Robert Hooke. Robert Hooke first observed it when he was observing a thin slice of a curved cell in a microscope. That means one empty space was surrounded by certain structures, and they are nothing, they are only the cell walls. But later on, it was found that the cell walls are unique only to plant cells and they are completely absent in case of animal cells. So that was stated by Theodore Swan. Theodore Swan, that is, it is unique only to plants. Unique to plant cells. So from the time of Robert Hooke, when he considered that this cell is surrounded by a rigid cell wall that is completely non-living, that is almost inert. From that time, we are considering that cell wall is inert, it is inactive, but in the late part of 20th century, the available evidence compels us to consider that the cell wall is a living part of the plant cells and it carries out certain metabolic reactions. There is an article in Science in 1998 in the magazine Science, that is in 1998, where it is the title of the that one of the topics is that is when walls can talk. Plant biologists listen. When walls can talk, plant biologists listen. This is the article, and in one conference, nearly 400 scientists have proved that actually the cell wall is not rigid, or it is not inert. It is not completely non-living. It carries out also some of the metabolic functions. So we have to change our concept. Now, the cell wall, though it is present only in plant cells, there are also certain plant cells where it is completely absent. For example, if you take the gametes means the male and female gametes, you can tell the sperms and the ovum, then it is completely absent. Otherwise, it is usually the cell wall is present. It is present in algae, in plant cells, also in fungi, then some bacteria, and also in some protists. Some protists cell walls are usually present. Now, what is the if we take the different what are the different chemical composition of the cell wall? Chemical composition of cell wall, will find that the chemical composition of all these things, that is algae, plant, fungi, bacteria, they slightly differ from each other. Now if you take the first case of algae, in case of algae, usually cell wall is made up of cellulose, then galactans, mannans, and minerals of calcium carbonate. While in case of the higher plants, in case of higher plants, it is made up of cellulose, hemicellulose, pectins, and proteins. In case of fungi, it is usually the cell wall is made up of chitin, and this chitin is usually a polymer of N-acetyl glucosamine. N-acetyl glucosamine is a polymer, and short usually it is written as NAG. That means chitin is a polymer of NAG, N-acetyl glucosamine. And if you come to bacteria, usually the bacterial cell wall is made up of mucopeptide, or you can tell that is peptidoglycan. That means both peptide means protein, glycan means carbohydrate. But here our main discussion is usually mostly these plant cells. Now, what are the different structures and how they are present? How can it be known? It can be known if the maceration is done. Maceration. So, what is this maceration? Maceration means if you dissolve a tissue in some acids, then everything will be dissolved. And what are the substances present within the cell wall that can be easily noticed? That is known as maceration. That means soaking some cells in acids. I am writing it here. Soaking tissue in acids so that it will be dissolved. That is known as maceration. So through maceration, it was found to what are the different types of substances present in case of this cell wall. It was found that the cell wall usually consists of the first one is the matrix. Matrix. Now, what is a matrix? Matrix means suppose this is the cell wall. So whatever is present here in this in a amorphous form, that is known as matrix. This is the matrix. And in a matrix, what are the different substances that are present? Now, usually in the matrix, you will find water. Water is 60%. They will get that of the pectins. Pectins 2 to 8%, and also you will get glycoproteins. Glycoproteins 1 to 2%, and these glycoproteins are mainly of two types. It may be extensin, and the other is expansin. The expansin is recently discovered that it is present also in the cell wall. Now, what is the function of these two? Usually, this provides support to the cell wall, this protein, and this usually helps in extension of the cell wall. Extension of the cell wall. So these two are very important from examination point of view. In addition to that, you will also get hemicellulose, and that is present usually between 5 to 15%. That means these are the different substances that are present in this matrix. That means in a matrix there is 60% water, pectins 2.28%, hemicellulose 5 to 15%, glycoproteins 1 to 2%, and in the glycoprotein the two common very important examples are, one is extensin, which gives support to the cell, and expansin, which gives or which helps in extension of the cell wall. So all these things are there present in this matrix. Then the second one is microfibrils. The microfibrils, these are mostly the cellulosic fibrils. Cellulose fibrils. So inside this matrix, several cellulose fibrils are oriented in different way. These are the cellulose fibrils. These are the cellulose fibrils. Cellulose fibrils. So these fibrils are made up of cellulose. That means if this cellulose gives rise to micelle, the micelle gives rise to, that is a microfibril, or also it is known as elementary fibril. And the microfibril usually gives rise to macrofibril, or simply we call it as fibril. Simply we also call it as fibril. So usually 100 molecules of cellulose, they combine together to give rise to a micelle. 20 micelles, they combine together to give rise to a microfibril or elementary fibril. And 250 microfibrils, 250 elementary fibrils or microfibrils, they give rise to a macrofibril or fibril. These are the different fibrils that are present in the matrix. So the third one is depositions. Now, if the cell wall is mature and permanent, then there will be different types of depositions, and depositions may be lignin, cutin, suberin, or even it may be silica, depending upon what type of cell and where it is present. So in maceration, we have found that these are the different substances that are present inside the cell wall. So this is the chemical composition, and after maceration, what are the different substances and they are present in the cell wall. This is clear. Now let us discuss the structural part of the cell wall. So when we will discuss the structure of the cell wall, we will find that usually in all cells there are two cell walls, but in mature cells we will find sometimes there may be 3 or 4 layers are also present. Usually, if you draw the cell wall, this is one cell, this is another cell. Now this layer is known as primary layer, and inner to the primary wall there will be secondary wall is there. This is the primary wall, primary cell wall, and this is the secondary cell wall. And in between the primary and secondary wall, this part, this is known as middle lamella. This is known as middle lamella. Then in some cells, usually there may be another layer inner to the secondary wall, but it is very rare. So in that case, that will be known as tertiary wall, but it is very rare. Tertiary wall. Now we will discuss one by one. And the first one is the middle lamella. Now, middle lamella, it is the interface between two adjacent plant cells. It is the interface between between two adjacent plant cells. That means it acts like a cementing force. We can tell it acts as a cementing force, or we can tell that it is glues together. It glues together two adjacent plant cells. It is glues together. It acts like a cementing force, and usually it is made up of calcium pectates. This is very important for examination point of view. Calcium pectates. And sometimes also you may get magnesium pectates, but calcium pectate is usually more in number. And this middle lamella usually formed when the cytokinesis takes place during cell division. Now usually you might have marked when the fruit is raw, not ripen, it is very hard, because these calcium pectates, they are very hard. But when the fruit ripens, gradually it softens. That is the softening of ripening fruits. So when there is softening of ripening fruits, what happens? The insoluble calcium pectates are converted to soluble pectates, so that we get some juice from the fruit. Softening of ripening fruits, the insoluble pectates converted to soluble pectates, and this is due to the action of a pectinase enzyme. So that is why the ripened fruits are usually soft. So this is about the middle lamella, which is the cementing force between the two. Now then inner to this middle lamella, this is the primary wall, and then inner to that is secondary wall. If we will discuss it in a very comparative manner, your idea will be very much clear.